record to this computer. Okay, so I'm going to show you how to do this DNA replication, and you can use this video to walk you through how you're going to set up your model. The first thing that you'll do is find this roll of paper in your kit and unroll it, and this shows you the sequence that you need to make. And then, as you can see here, I'll play the video. You can see that I made it out of the foam bits, so you have it all ready and you have it in the right order, and you can put it right on top of the paper because it's the same size, so you can kind of keep track of where you are. So that's our first step, and once you get all that done, then you can move on to the next part. So let's pause the video now, and you can go ahead and put all yours together and make them. And then once you're done, you can start the video back again. So go ahead and pause now. Okay, so now we will look at my chain. Yours should look like this. And you can see that the arrows are pointing opposite directions. That's the anti-parallel configuration of DNA. And then you have the little nubs in the middle that connect the two sides. And so once you have that done, you can get out your little placemat. This is a little helicase enzyme, and this is the enzyme that actually cuts that strand in half. These bonds that are in between each nucleotide, those are hydrogen bonds, so they're really, really weak bonds, and they just unzip like a zipper, and then they zip back up when they're done. So the helicase is basically like the little zipper pull on a zipper, and it's going to unzip them and leave two single strands of DNA. So I'll play that. While I'm playing this, I want you to do it too. So you should be sliding your DNA sequence through the helicase enzyme and splitting your sequences. So go ahead and do that now. And you're not going to split it all the way. Just split it until it's at this point, until your green piece matches the little 3' piece. That's kind of where you'll stop. And then we are going to start adding pieces in. So in comes, so when you're like basically at this point, you can add one of the DNA polymerases. You have two of them because you're going to have a DNA polymerase on each side. And you can try to figure out where the other one will fit. But here on this leading strand is what we call it. You can fit the first DNA polymerase, and it's kind of right side up. And what's going to happen is a nucleotide that matches that green one is going to come in and bond to the green one in that DNA polymerase. So the DNA polymerase kind of finds the right nucleotide, and then it binds them together. So we'll see that coming in. There's one that matches, and we're just kind of matching that up and building the other strand. And so pretty soon we're going to have two identical strands of DNA. At the same time, even though I don't show it here, at the same time we have the other DNA polymerase coming in onto the lagging strand. And this one is upside down because those two DNA strands are anti-parallel and they're opposite each other. So you're going to bring in your little DNA polymerase, pause the video and do that. And then you're going to start adding nucleotides in the same way, but you're going the opposite direction. So the nucleotides were being added towards the helicase on the top strand. On this strand, they're going to be added away from the helicase because they're opposites. So here we are adding those nucleotides, and it's kind of dragging along. Off it goes, and you finished, and now the DNA polymerase ran out of stuff to make copies of. So what has to happen is your helicase has to unzip some more, and then your DNA polymerase kind of like leapfrogs back to grab another piece of the DNA strand to start replicating. So go ahead and take off your DNA polymerase, and hopefully you have both strands with their little bits of copies. And then we will work on unzipping the helicase even more. Here we go. So we're unzipping, go ahead and do that. We unzipped so that we had a little bit more space. And if I had my act together, I would show that the DNA polymerase was still on the top strand too, and it was still adding nucleotides as it was running down towards the helicase. So as the helicase is unzipping, the little new nucleotides are coming in. But here's the DNA polymerase on the bottom, and it came back, it leapfrogged back to like right up against the helicase, and then it's going to start moving out again from the helicase. So here it is. So it's building little pieces, and then it's going to kind of join together where it stopped before. And this part here where you had two different chunks that were being made at 2 times, that is called the lagging strand, because it like kind of takes it a little bit longer because that DNA polymerase has to keep jumping back and restarting as the helicase is unwinding. The other strand is the leading strand because the DNA polymerase can just stay on there and keep bringing in nucleotides the whole time. And there you can see the DNA polymerase on the leading strand at the top. And you can see that it just continues to go in the direction of the arrow there. It's going towards the helicase on the top. And then the bottom DNA polymerase is going away from the helicase on the bottom, and then it has to keep leapfrogging back to do more. So make sure that you're showing that process and going through that so you can understand how this process works, and then you can answer the questions better. And once it gets to the end, you would see 2 separate strands that are exactly the same, that were identical to the original strand, except now you have 2, and it's a photocopy of it.